Good morning, Honorable Show Laureates, distinguished scientists, forum participants, ladies and gentlemen. Welcome to Day 2 Plenary Session of the Hong Kong Laureate Forum. How are you today? Well, that's the spirit. A very warm good morning to everyone. My name, once again, is Laura Kosensen. It's my pleasure to be the MC of the Hong Kong Laureate Forum. On behalf of the organizer, a very warm welcome to you all for joining us here in person, and for those of you who are joining us online through the camera screen, good morning and welcome. Yesterday, do you remember what we did? Yes, some people are nodding. We had a fabulous grand opening ceremony, and we also had 2 masterclasses on different disciplines. And I'm sure for those of you who have attended, you would have learned a lot and been inspired by our speakers who are all renowned in their respective fields. Today, we're honored to have 3 renowned speakers in life science and medicine amongst us to share their knowledge and research experience in which they have spent many years to study in various biological mechanisms that threaten human health. And in fact, their related studies have been used to explore possible treatments to not only save lives, but also enhance the life quality of human beings, Homo sapiens. Well, now I would like to introduce them to you one by one. Let's give them a warm welcome when I announce their names. First off, please join me in welcoming amongst us today Professor Chen Y.E., Pro Vice Chancellor, Vice President of Strategic Developments, and Li Ka-ching Professor of Biomedical Sciences in the Chinese University of Hong Kong. Professor Chan is also a council member of the Shaw Prize Foundation. Welcome, Professor Chan. Thank you for joining us. We also have this morning Professor Michael Welsh. Professor Michael Welsh is Roy J. Carver Chair in Biomedical Research, Professor of Internal Medicine, and the Director of Papa John Biomedical Institute in the University of Iowa. Welcome. Thank you for joining us this morning, Professor Welsh. And coming all the way from Beijing, right? 昨天晚上，今天早上到的 Professor Xiaodong Wang, Director and Investigator of National Institute of Biomedical Sciences in Beijing, joining us for this morning's panel session. Welcome and thank you for being with us. Well, upon completing the 3 presentations respectively, as we mentioned just now, there will be a Q&A panel discussion hosted by Professor Chan, and the audience's questions are more than welcome. So stay tuned, make notes, drop down questions, and in the Q&A session later on, do post questions to our panelists, either through raising your hands or through the HKLF app. Without further ado, ladies and gentlemen, I would like to invite on stage Professor Chen Y.E. to deliver his keynote speech on the topic of recent developments in the studies of genomics, cellular mechanisms, and protein functions. Warm welcome to Professor Chan.
Well, good morning, everybody. I would like to thank the Laureate Forum for inviting me to lead off this discussion. So what I have planned is to basically give you some idea of recent developments in the studies of genomics and cellular mechanism. So we all know that the breakthrough, in fact, in biomedical sciences in the last 2 decades is actually the launching and completion of the Human Genome Project, which starts off in 1990 and end up in 2003, when the human genome sequence was completed and announced. This project, besides the human genome, actually is important for biomedical research in general, because it did bring along a lot of new technologies that are being used nowadays, in fact, enhances the development of that area. One of the key technologies that is actually very much because of the Human Genome Project is the sequencing technologies, which allow us to look at our genome and also doing it very fast. As you all know that it takes 2 decades, well, one decade at least, more than 1 decade, to complete 1 human genome. But now, in fact, we can finish a human genome in actually less than half a day. So the evolution of sequencing technologies goes from the first generation. When I was a graduate student, I used Maxim Gilbert, sequencing, chemical sequencing, and then Sanger, chain termination. And in fact, nowadays, we can actually, at that time, the read length is about 300 base pair to up to a 1 kilobase is very long already. Now, in fact, one can do tens of KB fragments in a very short time, the long-read sequences, which is very useful. And because of the advances in sequencing technology, there are a lot of various approaches can be used now to do genome analysis, as listed in this part of the slide. We can actually generate data about point mutations, small indels, etc. And then transcriptomics, looking at expression, gene expression, and epigenomics, as well as metagenomics, etc. And all these technologies can be used, can be applied, and in fact, because the high throughput technologies now allow us to have comprehensive studies of biological molecules in a large scale, and facilitate, for example, functional effects of mutation, network pathway analysis, as well as integrative analysis. So those are the major technologies that we know and see that is brought about by the human genome technology. Another technology which is being used very often now is the so-called genome editing by CRISPR-Cas9. CRISPR means cluster regulatory interspace short palindromic repeats, which is actually a bacterial immune system that was utilized now for us to use in the lab and also work on human. In fact, the Cas9 is an enzyme, these endonucleases, that creates a chromosomal DNA and introduce double-stranded breaks, and the repair of which can actually be used to target genome modification. And in fact, because of the application of genome editing by CRISPR-Cas9, it actually revolutionized the field of genetic engineering. It has the potential to treat genetic disease, create genetically modified organisms, manipulate cell fate, and enhance the understanding of gene function, which you will hear more in the later during this session. And then another advance, which is also very used now, very often now, is the single-cell sequencing, and also spatial multi-omics. Single-cell genomics, single-cell sequencing, in fact, analyze the genetic information of individual cells rather than the entire population. So that, in fact, allows us to uncover the cellular heterogeneity and better understand the dynamics of cell fate determination, and also identify new cell types, studying disease processes at a cellular level and response to treatment, etc. That, in fact, is particularly useful in studying complex tissues where many different cell types are there. And advances in the single-cell RNA sequencing allow tracking of gene expression of individual cells over time. In fact, it allows, for example, like first identify cell types, and allow us to identify cell hierarchy, reconstruction, as well as inferring regulatory lacquers because of the single-cell sequencing. And together with advances in technology to identify different cell types, there comes the spatial transcriptomics, which allows the measurement of all the gene activity in the tissue sample, and so where the activity is happening. So advanced microscopy and cellular imaging is playing a very huge part in the advancement of biomedical research nowadays. Because of advances in imaging technology, such as live cell imaging, and development of super-resolution microscope and cryo-electronic microscope, which actually changes the field of cell biology by allowing us to visualize cellular processes at the molecular level, coupling with single-cell sequencing. So that allows us to study cellular dynamics and interaction with high precision. For example, if you look at the B, panel B, for example, which is a brain organoid, in fact, one can see the neurons that shows the green fluorescence, the green color. You can see where the neurons are, and also you can see the M-cherry labeling in the GFAP cells after viral transduction. So that allows us to look at individual cells where they are, and with the single-cell sequencing, in fact, we can see what activities those cells are doing. Another very important advances in cell biology is the production of, is the ability to, in fact, convert somatic cells into stem cells, so-called induced pluripotent stem cells, that was, in fact, initiated by Professor Shinya Yamanaka using 4 factors into mature somatic cells and allowed the cells to actually differentiate into stem cells, the so-called induced pluripotent stem cells, which, in fact, then can differentiate into various cells in our body. That, in fact, enhances the development of regenerative medicine and also allow us to use the technology to study pathology, toxicology, as well as drug discovery. So, besides the stem cells, now, in fact, another technology which becomes very useful is the so-called 3D bioprinting of tissues. If we can, by isolating the cells, we can actually reconstitute the tissue using bioprinting by putting cells onto scaffolds, and then you can use the scaffold with cells for doing transplantation as well as drug discovery and also do in vitro disease studies. In fact, now we can do bioprinting to generate, for example, like skin and cartilage, which is 2-dimensional tissues, as well as hollow tubes, like the vasculature, aortic valve, trachea, and also in solid organs, the so-called organoids. And in fact, we can also engineer organoids and use it to mimic a specific organs and use that for example, to use for like drug screening, disease modeling. And the major organoid generation approaches include using somatic cells by reprogramming it to iPS and induce it, or using embryonic stem cells or using adult tissues, stem cells isolated from adult tissues, and use that to generate 2-dimensional cultures as well as 3-dimensional cultures of the organoids. And the organoids can be used in a lot of ways. For example, like disease mechanism, use it to study disease mechanism, test drug toxicity, developmental and stem cell biology studies, as well as regenerative medicine, drug discovery, and also study infectious disease. Now, in fact, the ultimate goal of the Human Genome Project at the time was, in fact, to be able to treat disease eventually at the end. And one type of disease which hopefully will be very much helped by our understanding of the genome is, in fact, gene therapy. So this represents a basic approach for gene therapy, is to replace the mutated gene using normal copy of the gene. This is the original thoughts of doing gene therapy. But in fact, when you hear later our talk, one of the talks, you know that, in fact, this is not the only approach. Now there are a lot more approaches for gene therapy. It can be ex vivo genome editing for doing gene therapy. For example, on the left-hand side, one can actually extract stem cells or progenitor cells from the body, from the patient, and then manipulate outside the body, doing, for example, like genome editing, and then put the same cell back to the patient, the patient's own cells, to, in fact, achieve the therapeutic purpose. Or using viral vector, or lipid nanoparticles, in fact, to transfer the right cells or right genes into the body, to the patient. So this is another approach. There's also a very much used technology, especially because of the pandemic, is the RNA-based therapeutics. Now one can use small RNAs to treat disease, for example, by doing silencing, by doing mRNA drugs, which can in fact generate antigens for immune response, or replaced of the mutant proteins, etc. So this is another approach which actually come on because of the Genome Project researchers. And then definitely also using stem cells for treatment. Stem cells, embryonic stem cells, or induced pluripotent stem cells to generate cells of the different origin, of the different cell origin. Ectodermal cells, neuronal cells, or mesodermal cells, like adipose tissue, or endoderms, like the pancreatic islets. So all these, in fact, can now be used to do treatment, which is actually becoming more and more useful and more popular. So this is all I plan to talk because I think my time is almost up. So I do like to thank the Laureate Forum to give me this opportunity to share some of my thoughts with you all, and also the other supports that I get from the Hong Kong government as well as from my university. So thank you all.
Thank you so much, Professor Chan. Thank you. Please do take a seat off stage. Or just a question to the audience. Have you started marking questions? Like what are the possibilities? What are the developments? What are the application areas, etc.? Yes, I see nodding. Very good. Well, up next, I would like to have the honor of inviting on stage Professor Michael Welsh, who will be sharing with us on the topic of pursuing cystic fibrosis. Let's give a warm welcome to Professor Welsh.
Thank you, Professor Chan. Thank you, Professor Chan. It's an honor to be here, and thank you for coming to this. I wanted to talk today about advancing the slide. First, I want to say thank you. Thank you to be here. And so today, I wanted to talk to you about the arc of discovery leading to new treatments for genetic disease. And I want to do this in the framework of cystic fibrosis. It's perhaps the poster child of how you go from the bedside to the bench and back to the bedside. And in doing that, because this is a forum where there's lots of trainees, I wanted to particularly emphasize as I go through this, some of the events and processes that were important to me. There's many ways to do well in science and medicine. And this is some of the examples for me. They'll be different for different people, but I'll give you some of the examples. So how did I get started? As a third-year medical student, I saw a patient with cystic fibrosis in the pediatric clinic. She was probably about 7 years old or so, 7, 8 years old, and I remember she'd be coughing as we were talking to her. She was using her neck muscles to breathe because she was short of breath. She spoke in short sentences because she's short of breath. It was the first time I recognized the smell of Pseudomonas aeruginosa, characteristic smell. What was sobering was afterwards, as we left the room with this little girl coughing, we talked with the faculty and we learned about cystic fibrosis. And it was sobering to think that she probably was not going to live to her teens. And if she did, she was certainly not going to get out of her teens. And so then I learned some about cystic fibrosis. It's a multi-system disease. It affects the airways, liver, pancreas, etc. But when you gave the diagnosis of cystic fibrosis to someone, what you were telling them is that lung disease was going to be the major factor that limits their life. This is a lung from someone who had a lung transplant with CF, and you can see all this area here full of infection, basically pus, mucus and bacteria and pus. And then, as you read about this, invariably, the first sentence out of the textbook would be, cystic fibrosis is a lethal genetic disease, and it's autosomal recessive genetic disease. So I sort of filed this away in my memory. At that point, I did not begin to work on cystic fibrosis. But when you're a physician, there are certain patients that make an impact on you, and you don't forget the rest of your life. And that's what that little girl did. So at that point, I went off to begin finish medical school and then internship and residency. And I was impacted by my mentors. And I have a variety of mentors. Here's 6 of them that are on the wall in my office. And so for you students, postdocs here, you're thinking about mentors. And I think one of the important things about mentors is it's good to have a mentor, but it's way better to have more than one mentor. And when you look at those mentors, they're not all perfect. Nobody's perfect. So pay attention. Some of these people you want to emulate. You want to do what they do and follow their path. And some of them you want to say, boy, I sure don't want to do that. And pay attention to that. And don't do that in your own life. So pay attention as you go along to people that you want to mimic or follow their path and people that you don't. And I set off to do that. And I went to UCSF. And at UCSF, I began to study this problem. The problem of backwards. No, there we go. I went to UCSF, and I began to study this problem. Here's the airway epithelium, the lining of the bronchial tubes. Here it's on the trachea. And I became interested in this problem. How does the salt chloride, go from one side to the other? And this was driven basically just by curiosity. I just wanted to understand how this worked. And while I was there, I was working really hard, and I began to realize I didn't know anything. And after a year. And so I thought, I need to find a lab that's doing the latest thing, the theoretical aspects, and as well as the technical aspects. And so at that point, I decided to move to University of Texas at Houston. Now, if you look at this, you say, well, what is he doing? Moving from UCSF, right? But many people in this room will know what a fantastic university UCSF is. And you think about University of Texas at Houston, you think, well, that's okay, but it's not in the same scale. The point here I want to make for you is, don't let someone else determine your priorities in life. Go where you want to go. Don't let someone else decide what is excellence for you. Don't let anyone else determine your directions and priorities. Take it in your own hands and go forward. And that's what I did. I went off to University of Texas at Houston. And so you should consider moving when there are opportunities to move. And don't let names influence you. Go where the opportunity is. So at the University of Texas, I couldn't study lung anymore. I decided I'll study gut. I'll study the intestine. That's okay. I just want to learn how these people think. And that's what I did. And when I was there, I became obsessed with a scientific problem. I saw this phenomena. I was doing what I always called my midnight experiments, the kind of experiments where I couldn't give you a good rationale why I did it. I just wanted to see what would happen. Those kind of experiments are okay sometimes too. And I saw what happened one night, and I went and did the experiment several times, and I went home at 4 in the morning, and I couldn't sleep because I was thinking about it. And over the next months, all I could think about was this. If I was eating, I was thinking about this. If I was at a movie, I was thinking about this. If I was in the grocery store, I was thinking about this. I became totally obsessed. It's like this. Oops, excuse me. That's coming in a minute. Well, anyway, I became obsessed with it. I hope you can become obsessed with something so much that you just can't stand not knowing. And that, when I figured this out, I also had this incredible feeling of peace. I felt everything was smooth, and I understood something. In the scheme of things, how important this was, probably not that important, but it was really important to me, and I felt incredible peace. And at that point, I knew there was nothing else I could do with my life. This was going to be my life's work, to go this way. So part of this is, I had it easy because iPhones hadn't been invented yet. You have this problem. You have iPhones. Don't play with your iPhone. When you're in a lecture, don't be looking at your iPhone. Be thinking about what question you're going to ask the person who's presenting. Put yourself in the moment. That's where you want to be. Immerse yourself in the moment. If you're in the lab working on a problem, it's easy to escape the difficulty of wrestling back and forth with a problem by looking at your iPhone. Don't do it. Put your iPhone away. You can put your other brands of phone away too. Go all in. Go all in on this. So, and then from the University of Texas, I moved back to Iowa, and at Iowa, I continued on this problem. And we were investigating this, and then I read a paper by Paul Quinton. And what he said in this paper was that he said that there was well, rats. There we go. Abnormally low chloride permeability in cystic fibrosis. That was really cool because that fit with some of the things I was doing. And with that, I then collaborated with Jonathan Whitcomb. Jonathan had developed some of the first methods to culture airway epithelia. He was at UCSF. I had worked with him there. And what we found was that in the, what we found was that in the movement of chloride across this epithelium, there's some problem with chloride leaving the cell. And that was our observation. And that was important because it began to provide a unifying hypothesis for the pathophysiologic defect. You got a defect in chloride transport in the sweat gland duct. You have one in the airway epithelium. Soon we knew about this in the pancreas and in the gut. Had a unifying hypothesis for what was going on. And that set the stage for future work. And then the next important thing that happened was in 1989, Lap-Chi Choi, Francis Collins, and their colleagues identified the gene that's mutated in people with CF, and they called that CFTR. That raised the question, could you put CFTR, a normal copy, a wild-type version, into CF epithelium and correct the chloride transport defect? It took a long time, almost a year to be able to do that for reasons I won't go into. But then one Sunday, my postdoc came out to my house with a gel like this. And there it was. And we were so excited because that meant now we could go. The next day, I went to my chairman and I said, I need to take a sabbatical. And he said, when? I said, tomorrow. And then he says, well, where are you going to go? I said, my lab. And that's what I did. I went all in on this right away. And an example of that is this is the first paper we published on that. And what I want you to notice is two things that will be important for you. One is, I thanked, I'm having trouble with this. One is that I thanked Jim Hunter and Bob Fick, because I was supposed to go on the intensive care unit. That's to attend on the intensive care unit. That's a full-time job. And if I was doing that, I was not going to be in the lab focusing on this problem. And they stepped up immediately. So go to an environment where people care about what you do. And my colleagues here were primarily clinical colleagues, but they were interested in the whole endeavor going forward. The second thing was I thanked the people with CF, because that's where we got our cells. And they gave us those to help our work go forward. So how did we go about doing this? Well, we took cells, CF cells, and put in a fluorophore. Roger Chin, who you saw on Professor Chan's slide, had developed a variety of fluorophores. We put fluorophores into the cell, and then we loaded those cells up with iodide. And what iodide does to this fluorophore is quench it, so the fluorescence goes down. And then we used, I had no idea how much CFTR I would need. We got our hands on a vaccinia virus system and developed that because we wanted as much expression as we could get in the cells. We got the expression in the cells, and our idea was that if we did that, we would, whatever this was, whatever mechanism, see if it was in cells to allow anions, chloride or other anions to escape, that would somehow do it. And if iodide left the cell on that system, then fluorescence would go up. And here's some of the examples. This is some CF cells, and in these cells, we are expressing CFTR with the common CF mutation. And we look early and late, and what you can see is a little dimness there, not much. If we did this with wild-type CFTR, you can see that some of these cells were lighting up. And this was so exciting. At this point, I was doing these experiments myself. My postdoc was getting all the cells ready, expressing the right systems in it. I'd take the cells, and then I'd go into this little room with the spectrophotometer that we were developing. I was trying to develop this assay. So I'd disappear in there all day. And at the end of the day, I'd come out. and I'd started to see something. And I'd go around the lab. Well, look at this. Look at this. Look at this. And everybody in the lab would sort of look at it and they'd go, you could tell they couldn't see what I was starting to see, and they were skeptical. And then one day, I'm doing my experiments, I'd go to the incubator, I'd get my cells out to go into this little room, and it didn't say CF, mutant, and wild type. It said A and B. My postdoc decided to blind me without even asking me. And that was great. It was great for two reasons. First of all, I never missed at that point. You know, I always could tell which was which. But it was creating the environment I wanted, where everybody had a license to contribute. Everybody had a right to, was encouraged to participate, to be critical, to challenge. That's where you want to go. You want to go into labs. When you have your own labs, you want to create that environment. It's hard because the further up you go, people expect you to know. But you know, you don't always know. You want people to challenge you. I have benefited so much by surrounding myself with critical people, people who challenge me, people who tell me I'm crazy. You have to, that's the environment you want to be in for your careers. So, I guess that's what I just said. And once you do that, give credit to those people because they're really important to this overall endeavor. So what that did is it said expressing CFTR and CF epithelium corrects the chloride transport defect, expressing the common mutant did not, and it indicates that a fluorescence-based assay can report CFTR activity. Well, what does CFTR do? They called it cystic fibrosis transmembrane conductance regulator. What an awkward name. It had that name because no one knew. And what was the main ideas? Well, it was an ATP binding cassette transporter. This is a large family of pumps, things that pump, pump molecules out of the cell, into the cell. It's a huge prologous family of proteins. And so people figured CFTR must be some kind of pump. Here'd be sort of the engine down here that used ATP, and then some kind of pump, and then it had this other region called the R region. And people came up with all kinds of schemes for how this pump might control something over here. If you read the news and views and in Nature and the perspectives in Nature, they're full of this idea. Well, of course it can't be a channel. It must be some kind of pump. And how the heck does that pump work? I had been studying this. And so I knew that there was some problem here. In fact, everybody knew because we'd published it all, in Nature and Science. And so I put the simplest hypothesis. CFTR is a channel. That's the easiest way around it. And the point here I want to make is don't get contaminated by the prevailing wisdom that's not supported by rigorous data. Don't be afraid to challenge these things in your career. Remember this, Mark Twain said it. It ain't so much the things we know that get us into trouble, it's the things we know that just ain't so. And so this is don't be afraid to challenge the existing paradigms, the prevailing wisdom. So we went out and studied this. We made some guesses on if this was an anion channel. There must be some charges that are influencing that. We went about studying that, and we decided to change. We made our best guesses, and we decided to change some of these positive charges to negative charges. And then the way we studied that is with the patch clamp technique. We took a variety of different cells. Here I show HeLa cells. We'd express CFTR in those cells. We'd come along with our patch pipette, perfuse the interior of the cells, and then we could measure the flow of anions going back and forth across the cell membrane. And when we did that, we could use different anions, chloride, bromide, iodide, and we could look at this type of current voltage relationship. And what this did is could tell us the relative permeabilities based on when they crossed the x-axis. So in this case, bromide was more permeable than chloride, than iodide. And then the second thing we did then is came along here and changed some of these positive charges to negative charges. And when we did that, we completely changed the anion selectivity of the channel from bromide chloride iodide to iodide bromide chloride. And we did this in a variety of different ways. I'm just showing you one experiment here. It began to tell us that this, in fact, was a channel itself. I still remember Matt Anderson running out of the patch clamp room. He wasn't walking out of the patch clamp room. He was running, saying, I can't believe it worked. I can't believe it worked. It was so exciting to see this. So we began to, we learned that CFTR is an anion channel, and then that began to link the genetics, the molecular biology, and the physiology. And it provided a strategy for discovering and testing therapeutics. How does CFTR work? I think I'm going to skip that because of time. We investigated how CFTR works, how it uses ATP to open and close, and that helped us in understanding the function of the different domains, and that ultimately helped in the development of new therapeutics. So how do CF mutations disrupt CFTR function? Well, the way we went about that is introducing CF mutations into CFTR and then discovering what goes wrong. So here's the model of the cell. We've got the cell. Here's the apical membrane. This would be the air side up here, the interstitial side down here. Here's the nucleus here. CF is genetic defect. So you could have some mutations that lead to defective protein production. The protein's never even made. One example of that is a stop mutation at 1282. Some mutations, the protein was made in the endoplasmic reticulum, but was defectively processed. So it was never, the quality control system prevented it from progressing onto the Golgi and the cell surface. That is the most common mutation, CFTR delta F508. Some mutations let the protein get out, escape, get up to the cell surface, but they had defective regulation, didn't open and close normally. Example there is G551D. And the fourth class that we reported at that time, these are the results of many different experiments, was defective conduction. Everything works, but it's just not a very good channel. Doesn't let ions flow through very well. Example there is R117H. And there's many, many different mutations that fall into these classes. So what this work did is describe how mutations disrupt CFTR. And that informed genotype, phenotype analysis. Why does one person have mild disease, one person have severe disease? It gave a blueprint for developing therapeutics. If you want to go about developing therapeutics, you have to think about what do you want to fix. Do you want to fix this type of mutation that has defective processing, or this type of mutation where you want to increase the opening rate? And then finally, can mutant CFTR be corrected? Well, here's an example. This is CFTR, and it's creatively called into three bands, band A, B, and C. This is the mature protein. This is the protein that gets out to the cell surface. This is protein grown at 37 degrees centigrade. Here's delta F508. This is, most people with CF have a delta F508 mutation, and what you can see here is the protein is missing. Now, we'd been studying Drosophila in the lab, xenopusoocytes, and we're well aware of temperature-dependent effects, so we began to change the temperature. And that's where we got the surprise. Here's at 37 degrees, you don't see any protein, but as you lower the temperature, you start to see the protein. And that was super exciting because it said that the protein was maybe escaping, and we also found that it produced chloride current. In addition, we found that we could develop second site suppressors of the delta F508 defect. What that did was, one point here I want to make. I'm showing you the sort of the end results. A lot of experiments led up to these. So my advice to you is do many experiments. You know, and then in doing those experiments, you'll figure out, you put them together, what is the real experiment that I want to do, and you'll refine it and get down to it. I sort of like this quote. This is from Samuel Golden. The harder I work, the luckier I get. And I've been pretty lucky, and I work pretty hard. And so that's my advice to you there. So these results said the defects in CFTR can be corrected. They said that distinct mutation classes require distinct fixes. Some mutations affect more than one class. Delta F508 inhibits escape, and even if it does escape, the gating is not normal, it's reduced. So where did it go from here? Well, these discoveries provided the knowledge, the reagents, the practical approaches, and importantly, the confidence to develop small molecule correctors of mutant CFTR. We can't put people in a refrigerator and fix CFTR by cooling them down. But it said that it was likely you could develop some kind of reagent, some kind of medicine that might do this. And my co-awardee of the Shaw Prize, Paul Niglescu, led a team at Vertex Pharmaceuticals that developed highly effective medicines. And they have dramatically changed the lives of people with CF. People with CF are no longer, within a year, the rate of lung transplant dropped by 75%. People are going out, they're getting married, they're having babies. Dramatically changed the lives of people with CF, way better than I could have hoped for. I'll also point out we're not done yet. Not everybody has a mutation that's accessible, and you can add, so here we're focusing on trying to address how does loss of CFTR cause disease, and we've developing new approaches, and one of those is with the first non-mouse models of a genetically modified animal from pre-CRISPR days. So in ending, I'm really lucky. These are my colleagues, people from my lab, working in the basic lab. And the important point here is connected very closely. This is the clinical lab. People in the clinical lab all up here. And there's some people going back and forth, some physician scientists, I think that's been really important for us. This is a picture of a little girl. This is Kay Van Thornau. She was one of our patients. This is when she was a little girl. Her father was very inventive. He created respiratory devices. This is something that he made to help her breathe mist. Here she is, a little girl. She participated in a lot of our studies. A lot of our studies not focused always on trying to do a new therapy, but just to understand the disease in human beings. And I remember one time I was sitting with her on a bench waiting to do a study. And I turned to her and I asked, "Why are you doing this?" And she told me, "I know this won't help me, but maybe it'll help somebody else." The most noble human aspect. Here's who it helped. Her daughter. Turns out her daughter developed CF. This is her daughter holding a picture of her mother. At the time that Kay was participating, we didn't know yet her daughter had CF. Here, her mother died, and here's her daughter. And there's a reason she's smiling. Thank you.
Thank you so much, Professor Welsh. Thank you. I think it goes without saying that the final anecdote that you shared totally touched hearts. And echoing the topic is not just the pursuit of your studies, but the pursuit of passion. Passion in the pursuit and pursuit of passion. I think we all learned something. Another round of applause for Professor Welsh. Thank you. Up next, ladies and gentlemen, it gives me great honor to invite on stage Professor Xiaodong Wang. His topic is regulated cell death programs controlling the aging of the reproductive systems. Let's give a warm welcome to Professor Wang. Welcome.
Very happy to be here. It's hard to follow the previous talk, but still, I'd like to share with you what my lab has been doing since I came here, received the Shaw Prize in 2006. Seems like a long time ago. And my lab actually been studying Hung cell dyes. And this is a cell dye of apoptosis. And in a nutshell, we want to understand what's the underlying biochemical mechanism. In the language of computer science, what are the resource codes and what are the programs that govern these cells when the cell decides life is no longer worth living. And they turn on this program and they go through this dramatic changes and they check out this world. So for the last couple of decades, my lab, as well as many others, has been figure out this biochemical program. The key is these group of enzymes now we call the caspases. And these are the proteases that normally dormant in the living cells. But when the cell receives death signal, either from outside, like tumor necrosis factor family of cytokines, or from inside, for example, like DNA damage. And these signals will transduce through a biochemical pathway leading to the activation of these caspases. And then these caspases will be able to cleave many important intracellular substrates that will give you the cell death you just saw. And in this pathway, I just want to draw your attention to this particular pair of protein interactions. And one protein called SMAC that came out of mitochondria when the cell undergo apoptosis, and they will interact with another group of protein called IAPs for inhibitor apoptotic proteins that are able to inhibit the activation of caspases. And so in this way, the SMAC protein actually remove an inhibitor of apoptosis. I also want to mention, it's this little discovery that we made many years ago that we realized a protein normally functioning in the mitochondrial electron transfer chain cytochrome C actually is a double agent. That when they came out of mitochondria, re-trigger cell death by activating this particular cell death pathway. And it is for this work that we were awarded the Short Prize. So this is the molecular mechanism, how the SMAC interacting with IAPs. And this marshmallow-like is representing of IAPs, and this red stick-like is the SMAC protein, the part of SMAC protein interacting with IAPs. And you can, from the first glance, you can realize this interaction is rather limited to the N-terminal part of the SMAC protein. And it's only limited to this 4 amino acid of these N-terminal proteins. And by look at this and mimic this interaction, we collaborated with a group of chemists that we designed small molecules that mimic this AVPI of SMAC. And since the side chain of these amino acids are not involved in the interaction, we even able to derive modifications as well as linkers that to make these AVPI mimic protein dimer. The reason this dimer is good here is because the SMAC protein itself is a dimer. So by making an AVPI mimic and dimerize, we literally make a small molecule with molecular weight of less than 1,000 that can function as a SMAC protein, almost as good. But the only difference here is this SMAC mimetic can now penetrate the cell and they bypass the regulation imposed by mitochondria on the native SMAC protein. So once we have that SMAC mimetic, we start to play with it. And this will be a typical experiments we do in the lab. That is, we apply the SMAC mimetics to this particular cell. And of course, the cell doesn't care because this only eliminates the inhibitor of IAPs, but the caspase activation program has not been turned on. And so same thing happens if you treat the cell with a TNF-alpha. And even though the cytokines can turn on the apoptotic pathway from outside of the cell, but the program couldn't carry on because the cell has many IEP proteins. It prevents the program from turning on. But if you combine the two together, within a couple hours, more than 80% of the cells are died by apoptosis, as I just showed you. And we know it's apoptosis because this cell death can be totally prevented by adding another small molecule ZVAT, which is a potent caspase inhibitor, and all the cell will be saved. This is exactly like the diagram showed you. But what was surprising is we did the same experiments, and again, like just Michael says, and in a lab, you have many interesting people. Sometimes they give you a results that is perfect to fit the diagram, which is great. But then sometimes they give you results that is totally different than what the textbook tell you, which is a whole lot greater. And this is what happened. And again, we applied TNF and SMAC mimetic to these cells. And this cell is derived from human colon cancer. And again, neither of these treatments did much to these cells. But what was surprising was even you put these two together, you kill some of the cells, but the efficiency is much less than this cell. But what was surprising was now you put a you put a caspase inhibitor, ZVAT in. You did not even save of these cells, and you make the cell death so much greater, which was very strange and surprising. And this form of cell death not only do not need caspase, and the caspase inhibition even accelerated this form of cell death. And if I can show you, again, this is the cell death by in the presence of caspase inhibitor and TNF and SMAC mimetics. And clearly, this form of cell death is very different than the first one I showed you, which is apoptosis. But this form of cell death is just like popping a popcorn, and the cell membrane just popped. And this is not apoptosis, it's necrotic cell death. We then spend another decade and figure out how the cell dies by this necrotic cell death, now called necroptosis. And in this form of cell death, and it's carried out by this protein called MLKL. This is a pseudokinase. It has a kinase-like domain, but it does not have kinase activity. And upstream is a protein called RIP3. It's referred to as a receptor interacting kinase number 3. And the RIP3 will phosphorylate MLKL. Similar to caspase, the MLKL protein in living cells is dormant, is not active, but its activation is through phosphorylation. It's not through by cleavage like apoptosis. And we even mapped the specific phosphorylation site and developed a monoclonal antibody that is specifically recognize this phosphorylation events, and which give us the tools to probe in the disease tissues what happened when these pathways turned on. And the one first cases we did was we collaborated with an emergency room doctor who has many of these liver biopsies for people who get rushed to the hospital because they take a wrong medicine, they have liver damage, acute liver damage. And we probed this damaged liver biopsy, and we saw a massive activation of this signal, massive signal. And again, with this newfound tools, that monoclonal antibody that specifically recognize the phosphorylated MLKL, and we start to play even more, and we start to probe even more tissues. And this is one surprise we saw. And this is a tissue, this is a testes, and from mouse. And we probed the phospho-MLKL antibody probe on the 4-month-old testes, which clearly these are young testes. We did not see any phospho-MLKL signal. But when we probed the 18-month-old mouse, which will be, I would say, equivalent to human 70, and we see massive signal, phospho-MLK signal, and accompanied by this emptiness of this seminiferous tubules, which is where the sperm is generated. But if we knock out RIP3, we did not see any of this phospho signal, and we didn't see the thinner of the cell layers in the seminiferous tubules. We can even see still see the sperms in these old mouse. Then you may ask, these old testes looks normal, but it's really functional? And in mouse, we can actually do the experiments. And we paired with these old male mouse with two young female mouse, and we see, can they still reproduce? And here is the statistics, which was, again, quite surprising. And we used the 18-month-old mouse. And in the wild-type animals, only 3 out of 14 still generated a pup, and which is roughly about 20%. But if we use RIP3 and MLK knockout mouse, which otherwise looks perfectly normal, and even at this age, 80% of them still produce the pups. So not only their testes look young, they actually still functional. But then the question becomes, what's the catch? So the catch is we look at all the pups these otherwise wouldn't able to produce the pups, and all of them has birth defects. So which made us realize this program is in the testes to turn on when the animal gets reach certain stage to eliminate the old DNA from the gene pool. So that's the hypothesis. So our student finished this, write a paper and published, and he's ready to graduate. I said, you probably should wait for a few months to look at what happened in the females, because everyone will ask, what happened to the females? Before I show you the data on females, I also want to, you may also ask, all the studies in mouse, is that same thing is true in human? And of course, we didn't have access to the human tissues, but we're glad to see this paper, which was published a couple years earlier. And this is a group of physicians, and they have access to the old human testes biopsy samples. I figure this is probably from the prostate cancer patients. Again, this is seminiferous tubule staining with this phospho-MLK antibody. And again, you see this in 80 years old male, and you see this very strong signal in their seminiferous tubules. And they did even more strange experiments. I never asked how they get their protocol proved in their hospital. And they start to probe the semen samples as collect from different age of men. And they probe them with MLKL or phospho-MLKL. And also the number is small, so I would never use just 2. But I see, you can clearly see the trend. I would say right before you get your first faculty job, your MLKL and phospho-MLKL level is low. By the time you get tenure, and your MLKL and the phospho-MLKL seems to be, quite there. Then you win a Shaw Prize. And your phospho-MLKL in your semen, you just blow off the roof. And you need to do something about it. So clearly this pathway is also happening in human as well. So what about the females? Again, since we have these knockout animals, and we just check this is their ovary of young and old. and we counted their egg follicles number. And again, we see a very surprise results. And of course, you all know ovary is very, very different from the testes. And again, when the animal was young, at 4 months old, their egg follicle number is high and roughly no different between 3 genotypes. And when they are old, their follicle number is down, and in both wild type as well as MLKL knockout. But in RIP3 knockout, the follicle number remain high, and which is strange. Clearly tell you they are different than testes. And not only the egg follicle numbers, we also check the estrogen level, the hormonal level. Again, when the animal was young, the hormone level was high, and when they are getting old, the hormone level was lower. But what was interesting was in a RIP3 knockout animal, even when the animal reached 14 months old, their hormonal level didn't drop that much. So they seem to be young. Are they really young? Again, even for the females, we can do the experiments for every 4 months old young female and the 14 months old relatively old female, we pair them with a stud male. And we see whether they can still produce. And again, when the animal was young, 20 out of 20, 100% of them produce the pup. When they are getting old, the wild type, again, only about 20%-ish that produce the pup. And MLKL knockout, also roughly the same. But the RIP3 knockout, still almost 70% of them can still produce pups, even at this age. Again, you will ask me, what's the catch? The catch is the same. All the pup produced has a birth defects. So again, we believe there's another program happening in the ovary, and again, this ovary, this program, cell death program, get turned on to eliminate old DNA from the gene pool. And also, is that true in humans? And again, we collaborated with the OBGYN people, and we got some human ovary biopsy samples. And we first stained with RIP3, caspase-3, and we saw some of the caspase signal. And then we stained with RIP3, as I showed you in mouse. The RIP3 knockout will give you a phenotype. And we see the overlap of RIP3 and the caspase-3, which tell you that in ovary, there's apoptosis happening, and which is different than the necroptosis. And this RIP3 this apoptosis is RIP3 dependent. And again, since we are biochemists, we figure out exactly what happened to the RIP3 when the cell turned on this death pathway. And we found, again, it's a phosphorylation events that turn on this pathway. And we developed the monoclonal antibody that specifically recognize this particular phosphorylation events. And we saw this pathway is on in the MCS7 cell, which is derived from human breast cancer. And also, this cell is derived from human granular cells, which are the nursing cells from the ovary. And we probed this antibody with mouse, a different ovary of mouse of different age. And we can see this phospho antibody only pick up signals when the animal is getting old. When they are young, you don't see the signal. But with a regular RIP3 antibody, you see this like a bell curve. The RIP3 was expressed even when the animal was young. But I didn't have time to go through these details. That looks like when the animal was young, RIP3 was expressed in the follicles, granular in the follicles. But when this animal getting old, this particular antibody recognized the corpus albicans. So to summarize, that we think we figure out the two cell death pathways that seem to function in the reproductive organs. And one pathway is functional in the testes and seems to be conserved in human and mouse. And this pathway is a necrotic cell death controlled by phospho-MLKL activated by the RIP3. And in the ovary, and for the time's sake, I don't have time to show you all the data, we believe the prostaglandin 2 alpha is the key aging factor. And its level will rise in ovary when animal get old. And this will induce the activation of this particular pathway, which is an apoptotic pathway that activated by RIP3 and marked by the specific phosphorylation events. And this will cause the death of this luteal cells in ovary and contributing to ovary aging. And the work I presented to you is mostly done by the two graduate students, Meng Lingjun, who is the one that one day ran into my lab telling me these animals look strange. I said, what happened? I said, they doesn't seem to get old. And another graduate student, Li Dianrong, who worked out all about chemical details and of these two pathways I shared with you. And in our institute, and we have independent investigator like myself working on a variety of problems we encounter and we found interesting. And we also supported by all these core facilities that give us the tech support that allow us able to do these kind of comprehensive work with only a few graduate students. And to end, I'm also very glad that to tell you our youngest member of the principal investigators, who is born in 1994, have his undergrad training from Hong Kong Chinese University. And I'm hoping some of you in the audience will consider us as part of a suburb of Hong Kong in Beijing and consider your career develop once you reach there. I'll stop here. Thank you.
Thank you so much, Professor Wong. If we could have you to please remain on stage. Professor Wong, we would like you to join us on stage for the Q&A panel session. Please be seated. Well, I think you would agree with me that the three speakers have shared with us their research from a wide spectrum of scientific studies. And now we would like to invite on stage once again Professor Chan and Professor Welsh to join us for a Q&A panel session. Thank you, professors. Let's give them another round of warm welcome. Well, I hope you have your questions ready. If you have not yet submitted it through the app, you could raise your hands. Our mic runners around the room are more than happy to assist you. And with all that's all set, Professor Chen, the stage is all yours.
Okay, well, thank you. Well, thank you for the exciting talks. The past two talks are very interesting. And in fact, I'm sure that there will be a lot of questions from the audience. So maybe, okay. So there's one question there.
Thank you. I have a question for Professor Welsh. Great, inspiring talk regarding the mentor, and I believe you need to have as many mentors as possible. Not all of them can be your sponsors. So, but I have a question regarding the last part of your talk. There is a take after, so a good drug for almost all patients with CFTR gene deficiency or mutations, but do you think it's a miracle drug? Like, do we need other drugs? What will be the future? Like, such a great progress so far in this field, and Cystic Fibrosis Foundation support a lot of people who develop like new treatments, identify pathways. But what will be the future in this field?
Yeah, good question. Is it a miracle drug? You know, it feels like a miracle drug to me, because example is last fall, my colleague Tony Fisher, he's a pediatric pulmonary physician. He's taken care of a boy in a high school, and he's already had him in the hospital 2 times. That means he's been sick with lung disease. And he started on the medication, and Tony, my colleague, came to tell me he had just competed in the state cross-country meet, running. It's just unimaginable that that would be the case. So it does feel like a miracle drug, but it doesn't cover everybody. There are people with mutations that are not targeted. There are people who have adverse effects from it. There may be adverse effects we don't know. So where do we go from here? Number one, I think that it helps to focus on people who do not have mutations. They feel sort of left out. They're happy that other people have the medications, but they don't yet. So what can we do with them? They have mutations that can't be corrected. So there's a big effort focused on genetic treatments, gene therapy that Professor Chan talked about, gene editing. We initially, 20 years ago, did some of the first studies on trying to develop gene therapy. And we could get gene transfer, but it was never good enough to get therapy. And so that led to a lot of studies, and now it's back in the clinic again. I hope it will go forward this time. It's tough to get it to the airway epithelium. You'd think it'd be accessible and a ready target, but it's proved particularly challenging. But I'm hopeful that some of those approaches will go forward. Second kind of thing you could think about is other approaches. One approach that we're collaborating with a colleague at University of Illinois in Champaign is to put in a prosthetic channel. You know, if you lose your foot, you might have a prosthetic foot. Not as good as a real foot, but you can still get around with a prosthesis. So can we put in a prosthetic channel? And so we're working on that. We've formed a small biotech, and we've just finished phase 1 studies and headed towards phase 2. Is it going to work? It's too early to tell. Of course, we're optimistic, but I don't know. I think that with trying to approach this group of patients may then lead to treatments that'll be used for people with a common mutation, right? Let's say you develop a particularly effective and safe approach with gene therapy or gene editing. You might then move that to people with more common mutations. The other problem here, of course, is the expense. Gene therapy expenses are, our approaches, at least today, are incredibly expensive. Even the medications, the Trikafta, is very expensive. We need to think about how we can equitably translate these to the big number of people who could benefit but can't afford it. So a lot of challenges ahead, but I think it's a very exciting time.
Thank you, thank you. There's a question over there.
Thank you all 3 professors for your talks. My name is Rafael Lasso. I'm from the University of Toronto, and I study Parkinson's disease in mice. And I have a question for Professor Wang. And I was wondering, could you describe a little bit more about those birth defects in the RIP3 knockout animal-produced offsprings? Like, are we looking at defects that are more similar to what happens when, like, old human mothers give birth to kids and then there's trisomies or stuff like that? Or are we looking at like random mutations in the DNA? Thank you.
A great question. Which actually sequenced, all these sperms, old sperms, we actually surprised found we didn't find many mutations. That was the first hypothesis to check. But when we stain the old testes with the oxidative DNA marker, and we see the testes lining up massively. So to answer your first question, our hypothesis is that there could be a combinatorial factors. One thing I didn't show you is there is also defects in their seminal vesicles, and the decoloration and hypertrophy, and all the like human prostate as well. So there could be, we couldn't pinpoint exactly what's wrong that generate the birth defects. And the birth defect is, we didn't characterize them in details, but just from the first morphological glance, they look all different. It's not one thing wrong. It's like a lot of things was wrong. So it's just we just didn't really go into to characterize what's wrong with them. But since you are studying Parkinson's disease, what we are trying to do, we found the thing, and clearly, this program cell death contributing to this age-related tissue degeneration, and to make the old animal, testes looks young again, is not what we are trying to do. And even though human is probably the only animal species that choose whether to reproduce or not with their brain rather than their testes. But we also found this program cell death is also involved in other degenerative diseases. Like recent paper in Science, which looks quite promising, demonstrating this RIP3 MLKL pathway. It seems to be the primary reason for loss of neurons in AD.
Thank you. So actually, I have a few questions from the audience also sent through the computer. So the first question is that now that we understand the genes and the functional defects underlying cystic fibrosis, are we looking at completely relieving the deadly symptoms of cystic fibrosis in the next decade or so? That's for Professor Welsh.
Yeah, so the question is, are we looking at relieving the symptoms and defects everywhere? Likely, with Trikafta and with these medications, those medications should affect CFTR everywhere. But they don't. Mm-hmm. You persist with inflammation and infection in your lung, even afterwards. Your lung does better, you get better pulmonary function tests, you improved, but they persist. So I think there's epigenetic changes that persist. The cells are living a life influenced by the previous time they've had. And so I think we need to understand that. As we look towards gene editing or gene therapy approaches in the lung, then it's going to be really interesting to ask, well, what happens everywhere else? So we just completed a study with colleagues at UC Irvine looking at arteriovenous metabolomics and trying to understand how do the different organs function, what goes in, what comes out in terms of metabolomics. It's really fascinating. Led me to think about kidney in a way that I never thought about in cystic fibrosis. And that causes me now to wonder, if we fix the lung, but don't fix these other organs, what will be the life course in these people? I think we don't know that. And there's a tremendous amount to know yet. In listening to your talk at the start, I was thinking about one thing where we lag behind is in metabolomics. We can look at DNA expression levels on the single cell level. We've got tremendous ways of looking at individual cells. We lag behind in our ability to look at the metabolism of individual cells. I think that's a frontier where we need a lot of work yet.
Yep. There's a question there in the middle.
Right. Thank you very much for the very inspiring talks this morning. I have a more general question, which probably applies to most of us here. So I think we all agree that we want to work on important problems, but how do we identify which problem is important out of all the problems we have? And secondly, important problems take time to solve, but academia nowadays, we have the pressure to publish and publish regularly. So how do we strike a balance between working on something important that takes time and being productive enough so that we can stay in this route in academia? Thank you.
The questions are a lot more philosophical than science. Who want to take the first? Who want to take the first try?
Well, I can't answer how to tell what a specific problem is that's important. But in terms of academia, when I'm mentoring new young assistant professors that come in, I tell them, you know what? You don't need to cure cancer with your first paper. You can make an effort forward, and what that does is say, hello, I'm here, I'm alive, I'm at Chinese University of Hong Kong, and I'm publishing a paper. And that's your entree. In terms of the important problem, I'm not sure how. We listened to that yesterday in the forum after the first set of talks, and it was hard to come away with a specific answer. I think you want to work on something that you're interested in, that you're hot after. And sometimes that may or may not appear to be important at the time. Many of the tools that I use in my research wouldn't have been considered groundbreaking, but they aid the whole thing going forward. I don't know. I'm going to turn this over to my colleague here because he'll be able to tell you how to pick an important problem.
I heard this question a lot. My answer is, from a philosophical perspective, I think any problem is an interesting problem, is a good problem, as long as there's no answers. And if you can solve any of the problems that has no answers, making even a slightest progress in our understanding of an unknown world, I would say, you will make yourself a very successful scientist and make yourself very happy in solving this. But in a practical sense, even when I was an undergrad, I have a habit of writing down what is unknown. Like when I study, for example, cell biology, and I like to write down what is unknown. Later on, I may not able to work on all these, and maybe not working on any of these, but it's become a habit of trying to identify what is the problem still don't have an answer, and then you wait for your opportunity. And I've been thinking, I look back to the notes I wrote in college, and many of them I never get a chance. Almost none of them I got a chance to work on. But what's interesting was, many of the things I wrote down are now solved. By a variety of, by all these different scientists. So, making a habit of writing down and what you think is interesting,
And that will, the habit itself, will lead you to a problem that you may eventually work on.
I really like that answer. That's why. One thing, when I was talking about when I said I became obsessed, that was the entree for me because I knew I was going to work on this. The other thing is asking questions. Xiaodang was asking questions by writing this down in his notebook. And you all are curious. You certainly were all curious at age 2 when you were picking up everything you could find and putting it in your mouth and testing what the world was and feeling things and wandering around. And we don't want to lose that curiosity as we get older. Sometimes we're sort of afraid to show our curiosity because if we ask a question, somebody will think, well, they're not very smart, they're a little stupid asking this question. But pushing yourself to ask questions is really important. I learned that, particularly learned that in 2 ways. One, when I was a young faculty, I realized somebody'd be speaking about something and I didn't know the answer, so I'd ask the person next to me, did you understand that? No. Did you understand that? No. And then so I'd raise my hand, and I figure at least there's 3 of us. The second way I learned to do that is when I started to invite people to the University of Iowa as speakers. Well, after you've given a talk, you don't like it if nobody stands up to ask a question. So if I'm the host, after every talk, I would have 2 questions in my pocket. So if I stand up there, I'd say, any questions? If there wasn't immediately a question, then I would ask a question to the invited speaker to break the ice. But that taught me to begin to think about what are the implications of the work that's being described. I'd start to think about what are the implications of this? How would I do this? What does this mean? And that helped me a lot to go forward. So I'd encourage you to do that too. And I'd also encourage you to ask questions. I give you points. I don't know what your name is, but I give you points just for asking the question. It's a great question, but it doesn't even matter if it's great. It says that you have enough confidence to stand up here in front of all these people and ask a question, and that's really good.
Yeah, maybe I'll just put in my part. I think nowadays, you really have to pursue what you're interested in. Don't worry about like paper, grants, tenure. I think that's something which will come if your work is good. Really, if you are not interested in what you're doing, you're in big trouble. Because research is really a lonely journey. I mean, you see the people who are successful in doing research because they're interested in what they're doing. So I think that's something that you have to keep in mind. If you are in academia, for example, that really is the most important thing, I think. You have to work on something you are interested in. Okay, last question. Yeah. Yeah, the one, right. Sorry.
Thank you. Perhaps a question for Professor Walsh, but maybe applicable to the others as well. I mean, you've given a lot of lessons from your own personal journey, which I think will probably be very valuable for younger people. But would you be in a position to say anything more generally about the obstacles that you face in pursuing these very important questions? Obviously, each person runs into their own obstacles, but maybe do you have any general prescriptions? People will run into obstacles, and they have to overcome those. Any lessons?
Well, of course, there's obstacles all the way. And there's obstacles in science. There's obstacles if I've got a restaurant that I'm trying to run or a cement company I'm trying to run. Once you're going for this problem, if a problem you're identifying, I think the resilience is important, but also just finding a way around it. Sometimes I've banged my head against the wall and banged my head against the wall, and finally I say, you know what, I'm not going to get this. I'm going around and taking a completely different route around this. I should have a better answer for you, particularly since you're a young trainee. But maybe you have an answer.
I don't know whether I have an answer, but I do have a solution. You know, like I also get this question a lot. They said, you know, you must be confident. You know, you seem to have a success in early in your career, and I keep telling people. I said, you know, I almost have, you know, constantly panic attack. That worry that, you know, I'm not able to do, you know, what I have done before is all just pure luck, and I'm unable to do it again, and just I'm going to hit the wall, completely hit the wall, and I couldn't get out. You know, have this feeling all the time. But I do have a solution. You know, one is, which is all, it's not just the solution I practice. One is, you are not fight alone. And you always associate yourself with smart people. With smart people, with people who are encouraging, because in many cases, the solution is not obvious. But together, and through encourage each other together, we bouncing ideas, then we can have a start. And I have another solution which I practice, is I run. I run marathons, which I didn't start. You know, I don't have a body of a marathon runner, and you can see obviously. But I started running marathons, you know, a few years ago, and I do learn that no matter how impossible the obstacle seems, and the key thing is just to start. It doesn't matter how long is their journey, but once you start, you will get there. So that's how I practice.
Well, with that, I think because of time, we have to stop. But hope you enjoy the science as well as the wisdoms shared by our laureates. So let's thank them with a big applause.
And a big thank you to Professor Chan for chairing the panel for us. Ladies and gentlemen, let's put our hands together once again for Professor Chan, Professor Welsh, and Professor Wong. It's possibly the best panel that I have ever attended. Thank you so much, gentlemen. Thank you. Thank you. I would like to once again thank our panelists not only for their scientific sharing, but their authentic sharing. I know that there are still a lot of questions that you would like to pose. Nonetheless, the morning session have come to an end. So on behalf of HKLF, I would like to thank every one of you for joining us this morning.